Okay. So, good afternoon, everybody. My name is Maria Angelica Ouellet. I am the research scientist at the Lakeland Agricultural Research Association, located in Bonneville, Alberta. It is my pleasure to introduce you to Dr. Miriam Fernandez. She is a researcher at the at Agriculture and Agri-Food Canada at the station of Sweet Current, Saskatchewan. Miriam has worked as a research scientist for Agriculture and Agri-Food Canada at Sweet Current Research and Development Center in, of course, Sweet Current, Saskatchewan for over 30 years. She spent the first part of her career working on the impact of management practices such as cropping sequence, tillage method, glyphosate use, and climate change on crop development and health, among other responsibilities. Her involvement in organic research since the late 1990s led to the establishment of a multidisciplinary organic research program at Swift Current in 2007, with research being done since then in collaboration with multiple partners and the guidance and support of organic producers. This program has included a variety of different projects, with the main objective being the adaptation of different cropping strategies to our environment while making them more sustainable, resilient, and productive. She has published extensively in popular media and peer-reviewed scientific journals. So thank you very much, Miriam, for assisting to this webinar. And yeah, go ahead.
Yeah, thanks a lot, Angelica. Hello, everybody. I thank you very much, Angelica, for the invitation to present our research. I'm going to start right away. As organic producers or low input producers, I guess people are aware that there are several disadvantages of growing soil legumes for nitrogen supply. And they range from being negatively affected by the environment, either very dry or very wet. They are low competitiveness in general to weeds and other crops. They also decompose very rapidly. Therefore, they don't contribute much to organic matter. And most of them are also in the Western prairies are susceptible to root rot, fusarium, and aphanomyces. So crop production based on the intensive soil legume cropping might not be sustainable under a scenario of climate changing. And so therefore we need to investigate alternative practices with mixed crops such as cover cropping and intercropping. And they are both of them are important components of regenerative organic and low input cropping systems. And they have also been the focus of most of our research for the last few years. So as most of the people attending this webinar would be aware that cover crops are very important for a variety of different reasons, improvement of soil health in a variety of different ways, increasing biodiversity, crop biomass and yields, and suppressing weeds. Therefore, they help to reduce or eliminate the need for synthetic inputs. But there isn't much information on cover cropping in semi-arid regions in other parts of the world, especially under organic management. And there has also been the belief that some of these practices would not be successful in our semi-arid prairies, partly due to the fact that crops not adapted to this region have been tried and failed. So this cover crop project was funded, it lasted several years. It was funded by WGRF, Western Grains Research Foundation, through the Organic Cluster 3. And there were in-kind contributions from Imperial Seed and Fig Seed. So this project started in 2017 until 2023, and there were several people involved in our team in different capacities. And today, Noe Welchley, who is responsible for our current field operations, is attending, and he will post his email address on the chat so that if you have any specific questions about our operations, you can ask him now or later. So the main objectives of this project were to investigate the ability of diversified cropping systems that include full season cover crop mixtures, which we will refer to as blends, with different functional groups in rotation with grain crops. And our main objective was to determine whether they will increase soil fertility and organic matter, suppress weeds and diseases. And at the same time, we also, through this research, we were able to identify biocontrol microorganisms. And I don't think I'll have much time to talk about that, but I will likely mention it at the end. And also, and of course, improve the productivity and quality of the cash crops. So overall, we wanted to, our main objective was to overall improve the sustainability of organic cropping systems and increase their resilience to climate extremes. So this work is of value not only to organic and low input conventional producers, but also and not only in our branch also, but also in other regions in Canada or elsewhere where similar environmental conditions might occur, and which, as you know, are becoming increasingly common due to climate change. So just a few details about our operations, but again, if you would like to know more about them, you can ask Noe or myself. So we seeded the cover crops in the spring, and we mowed them at flowering. We had monocultures and blends or mixtures, and they were followed by a cash grain crop, which was seeded after spring cultivation. And each sequence of cover crop followed by durum was on a different piece of organic land, and all lands had been organic for at least 3 years. And we took multiple measurements throughout the growing season. This is a list of most of them. I'm not going to talk about all of them in this presentation. So if you would like more information, you can always ask us, but I will address some of these parameters. So, and you can, there are several articles that have been written on this research by us and by others, and you can find them in the Sask Organics website under current. And these are two of the latest ones. The one at the top is basically a summary of the whole project, and the one at the bottom is mostly adaptation of cover crops to the Western prairies, and this was published in the Abundance magazine of Sask Organics. And again, you can find them on their website. Just one sec. Oh, sorry, I couldn't. Okay, so we had a phase 1 and a phase 2. I'll just describe them briefly. Phase 1 was basically the phase where we had the blends and their respective monocultures, and phase 2 is where we had the durum wheat grown after the cover crop blend. So in every year we had the two phases present. And the reason why I crossed out 2020, that was the year of the pandemic, and we weren't allowed to seed the phase 1 that year. The phase 2, we were able to seed it because it was just durum wheat, but we weren't able to go ahead with the 2021 phase 2 because we didn't have a phase 1 in 2020. So this is a list of most of the species that we tried, and there were even some more that I didn't include here. The ones that I listed here were tried at least for, I believe, 2 years or so, and there were others that just did not work at all. So we didn't bring them back. So these are some pictures of the brassicas, butter radish, for rapeseed and turnip, oat and Japanese millet. And some of the legumes, these are the ones that grew the best: chickweed, vetch, crimson clover, and a fourth bale of Cecilia, which a lot of people, and I will be making some comments about that many people like it. So just to remind people of the kind of weather that we had since 2017, we had a lot of precipitation, as you remember, in 2016, but then we started a dry cycle that hasn't ended yet. So in this star here represents the location of our research center. So as you can see, it has been quite dry. So, and they, but even, under the dry conditions, we were able to examine and determine the performance of cover crops under not very favorable conditions, which was an opportunity that we wouldn't have had otherwise if we had started earlier. So selection of the most adapted species to our environment was mostly done in the first 2 years of this project. And that included not only their individual performance, but also how their growth was affected by the competitive ability of other crops in the mixtures or blends, so that one specific species might perform in a given way in a certain blend, but in a different fashion in another blend with more competitive species. And we also wanted to determine the most optimal number of crops in each plant. We, at the beginning, we tried even, and because it was recommended to us, up to 12 species, and we ended up with, I think it was 5 to 7 species. So what we did, this is from the first year, what we did, when we started assessing the suitability of the cover crops to our conditions. So here in the y-axis, we had wheat biomass and at the bottom crop biomass. So we wanted to know how they performed in relation to both parameters. So here on the upper left, you see the clovers, which had low biomass, but a lot of wheat biomass, so they weren't very competitive. And at the other extreme, you had Japanese millet and oat, which had high crop biomass, so they were really good, but and very few weeds. They were very competitive. And in the middle, we had other species, including the blends, which I'm going to be referring to in a minute. So therefore, this is the same list that I showed you before. The ones that I listed in red are the ones that we basically ended up discarding because they didn't perform as well. And I'm not going to talk about all of them, but for example, we discarded them for different reasons. For example, per millet was it was competitive, it was good, but it was very susceptible to leaf diseases, so we didn't want to bring it back. Hairy veg as you likely know, is a very good nitrogen fixer, but it basically became a weed, so we didn't want to bring it back. And Beta Faecilia, a lot of people like it, and we like it too. And it had good crop biomass, but it wasn't competitive at all. So, and of course, the clovers were quite a, sorry, okay, so I just wanted to mention in regards to the clovers, we tried different species, as you could see in the previous list, but unfortunately, most of the clover species that we tried under our conditions did not perform well due to the poor weed competitiveness. And this is a picture of Persian clover, which was a disaster. As you can see, it's just weeds. And but not all of them were that bad. The ones that we ended up selecting for our trials were crimson clover and subterranean clover. That you can see them here. So they are the ones that we kept for our trials. So in all years, the cover crop blends consisted of the three functional groups: brassicas, grasses, and legumes, but each blend focused on a different group. And in addition to that, we also grew each species as monocultures. So blend 1 was dominated by grasses, blend 2 by brassicas, and blend 3 by legumes, but all of them or most of them were present in all the blends. So as you know, crop growth depends greatly on the soil zone and environment, and that is basically what we focused our research on. But depending on the functional group and species, some crops can dominate while others would perform poorly. So, and so the growth of the less competitive species, including the clovers actually, in a blend depend on the competitive ability of the other crops in that blend. So the seeding ratios need to be adjusted accordingly. And so the clovers, for example, hardly grew in the presence of brassicas, for example, which are very competitive, but grew better when they were surrounded by less competitive species. So because of all this, we advise that if you are going to mix different cover crops, you use the expected crop biomass under your own conditions and don't rely on recommendations, which are usually based on other environments, and that you rely on preliminary testing and also on the forecasted weather if at all possible. So we recommend that producers that want to embark on this work that they do preliminary testing first, and before they design their mixtures. So here you have a. I don't think you can you would be able to see all the numbers here, but this is basically a list of the species that we had in each of the blends in the seeding ratios and rates in kilograms per hectare and pounds per acre. And so as you can see, we varied them. We ended up with this combination in 2022, which is the same one that we will use in the future. But we learned quite a lot from 2017 until now. And so we had to change the seeding rates accordingly. So the blend one was focused on grasses, the brassicas, the brassicas dominated in blend 2 and the legumes in blend 3. And as you can see, there were a lot of legumes that we included in this blend because of how because they are not competitive enough. So we ended up adding more legumes. So here is a picture of the blend 1 with grasses, blend 2 with brassicas, and blend 3 with legumes. And you can see here at the bottom on the right-hand side the clovers, some clovers growing under grasses. So I'm going to start with data on weed biomass, which of course is very important for cover crop work. So here, this graph shows wheat biomass here on the y-axis, and in all the monocultures here on the right and the blends on the left. And this is all in kilograms per hectare. So as you can see, the wheat biomass in the legumes, especially the crimson clover and the subterranean clover, even though they were more competitive than the other clovers, but it still had more wheat biomass than the other monocultures and the blends. But I will be talking about the blends in a minute. So, and the species that had the lowest amount of wheat biomass was actually 4-HP, was the most competitive. Legume oat was very competitive, and badger radish was the most competitive of the brassicas. So in regards to the crop biomass, this is the same graph as before, just showing cover crop biomass, which is taken at maturity. So here you can see that the highest biomass was observed in the grasses, both oat and Japanese miller. And as I mentioned before, and this was consistent throughout the years, started in 2017. This is what we noticed right away. And the lowest was, again, in the legumes. And for all the legumes, actually, not just the clovers, but the clovers had lower crop biomass than the other ones. So here I put together the biomass, the crop biomass at the top and the wheat biomass at the bottom. And as you can see, there is an inverse relationship, which you would expect between wheat biomass and crop biomass, so that the highest the crop biomass, the lowest the wheat biomass. And it works in most cases. So overall, brassica and grass monocultures had high crop biomass and low wheat biomass, and this was especially so for Baja radish and oat. And I thought I need to mention that the oat cultivar that we used was AAC or Avena, which you might know that is the first oat cultivar that was developed for organic production, although it's also suitable for conventional production. And for that reason, it is very competitive, and it also has a very good milling quality. So it's recommended for organic and conventional production. So in contrast, the legume monoculture had the lowest crop biomass and highest wheat biomass, especially the clover. So this is basically a summary of the graph. So if we put this in numbers, and this is the same data that I showed you before. And as you can see, the brassicas and grasses are not that different from each other. They are very competitive in regards to wheat biomass. And the crop biomass is also quite decent, especially for the grasses. Whereas at the other end, the legumes had the lowest crop biomass and the wheat biomass. But you can see more clearly here in this table that there are differences within each of the functional groups. For example, the colors here had high wheat biomass and much lower crop biomass, and that was the main reason why we ended up dropping it. So, there are differences in the among them. So, and Bacillus, as you can see, as I mentioned before, even though the crop biomass was okay, it had a very high level of wheat. So we didn't use it much. We only use it in some of the blends, but not much. And they compare the wheats and the fallow are very comparable to the legumes and the Bacillus because they are as high or higher than in those functional groups. But of course, the grasses and the brassicas perform much better. So, so we can then conclude this, and this confirms observations made by many other people that the brassicas and grasses are more competitive than the legumes. And one of the factors that contribute to their higher competitiveness is the allelopathy. And I'm sure you're familiar with this factor. And allelopathy, the chemicals that we are interested in are the ones that are leached. Oh, I'm sorry. Sorry, I think I missed that. Okay. Sorry, can you see it now or
Yes, we can.
Yeah, okay. So
Yeah,
I'm
just
sorry about that. Okay, so the allelochemicals that we are concerned about are the ones that leached from leaves through rain or fog or dew, and the ones that are released through decomposition of plant litter, and also the ones that are that decompose in the soil after harvest or a termination of the crop, and through exudation from the roots. So in general, wild types of brassica are they're most allelopathic ones, are more allelopathic than the domesticated brassicas such as canola, due to the presence of high amounts of glucosinolates. And domesticated cultivars um, don't have high levels of these products because they are bred out. So, but the degree of weed and pathogen suppression depends on the brassica species and cultivar because they vary quite a lot. But it's important to be aware that mulches could be a bridge for disease to the following crop. So if the cover crop species are host to the pathogens of the catch crop. And a good example are the brassica cover crops, which are followed by canola or mustard. They could increase the risk for diseases such as clapproot. So it's very important to be aware of that and to plan your rotation carefully. In terms of the grasses, rye is very well known for its allelopathy, and it has many different allelochemicals, and they're also effective against soil-borne pathogens and also nematodes. But rye cultivars differ in their allelopathic potential. And in general, concentration of the allelochemicals in rye is highest when grown under low to moderate fertility, suggesting that biofumigation might be most effective in organic and low input systems. And this is actually true of other inputs or the products such as biocontrol agents that are usually more effective under low nitrogen and than high nitrogen, and therefore are more effective in organic systems. At the other end, you have legumes, and they have some degree of allelopathy, some of them, but they're usually in general they are lower than for grasses and brassicas. And we also have to remember that legume crops, because of their nitrogen fixing ability, also have a stimulatory effect on weeds through the nitrogen released from their residues. And we did some work in the greenhouse actually some years ago that showed that hairy veg, and which is a very good nitrogen fixer, had a stimulatory effect on weeds more than other legumes. So that needs to be taken into consideration. So now I'm going to go to the blends, crop and weed biomass. And this is the same graph that I showed you before, but now I'm focusing on the blends on the left-hand side. And as you can see here, the blends had biomass that was comparable to the brassicas and grasses, and low biomass at the bottom, especially blend 2 and blend 1. So they performed very well. And that was also true of blend 3, as you can see, that was mostly legumes, but the wheat biomass was lower than the mean of the legumes on the right-hand side. So this is the same table that I showed you before. I just added the blends at the top. And as you can see, again, the mean of the crop biomass for the blends was comparable to the other groups, and the wheat biomass also was pretty good, even though blend 3 had somewhat higher wheat biomass than blend 2 and 1. So in summary then, the blend dominated by grasses and the one dominated by brassicas had consistently high crop biomass and lowest wheat biomass. And oat was, or avena was among the top biomass producing species in all blends, while the blends focused on legumes had the highest wheat biomass of the blends. And as I mentioned before, most legumes are less competitive against wheat. So in regards to the crop biomass, even though they had low crop biomass in the monocultures, they had similar crop biomass in the blend than in the blend dominated by grasses. And this could be attributed to the grasses, which constituted most of the crop biomass in blend 3. So if you remember the graph that I showed you before, you can see a similarity between blend 1 dominated by grasses and blend 3 dominated by legumes. And this has to do, this is the top is what I showed you before, the seeding rates for blend 3 throughout the years, and the one at the bottom is actually the yield that we had, the crop biomass. So, and you can see here that in general, we had the grasses had even higher crop biomass than the legumes in this blend. And this is the advantage and the need for having grasses in this blend, as opposed to having, you wouldn't be able to have a blend that is just dominated by 100% by legumes. It wouldn't work well for many different reasons. So adding grasses to it and works very well. So the last thing that I'm going to mention about the phase 1 is, and I won't have time to talk much about this, in regards to, we also saw some very encouraging observations in regards to root rot severity. Here you had the monocrop, chickling vetch in this case, and how root rot severity in this crop was associated or compared, I should say, to the other blends. And in blend 1, chickling veg had 31% less root rot. And here in 2018, blend 1 had 40% less root rot. And so this is the advantage. And this is something that we saw, and we have reported quite a lot on it in the intercrops, how grasses and especially the brassicas are able to control root rot, not only in when grown together with legumes, but also in the following cash cereal. So, and I should mention that most of the fungi isolated from the root rot affected roots were Fusarium species. So phase two is that we looked at the impact of cover crops and monocultures on soil nutrients and development of the following cash crop, which was durum wheat. So, and we saw that the impacts of the cover crops were carried over to the following year. And so I'll show you soil nitrogen the next spring, so measured before actually we seeded the cash crop. And I'll also show you wheat biomass in the following growing season and the growth, yield, and quality of the next crop. So in regards to soil nutrients, we found that nitrogen at all soil depths, from 0 to down to 60 centimeters, was significantly different among the monocultures and blends the following spring. But for other variables such as phosphorus, potassium, etc., there were no significant differences among them. So this shows the spring soil nitrogen in the spring in the different. This is basically the same layout that I had in the previous graph. So you can see here that the lowest nitrogen in the spring was after the brassicas. All the brassicas had basically lower levels, and the highest levels, which is not unexpected, of course, was after the legumes, including the clovers. And in regards to the blends, the blend, again, not unexpected. Blend 3, which was dominated by legumes, also had a higher nitrogen concentration than the other two blends, the blend with brassicas and grasses. So therefore, so as a summary, soil nitrogen was significantly lower after the brassicas than after the legumes, but it was highest after the legumes. And it was also higher after the blend dominated by legumes than after the other two blends. So what happened with the wheat biomass the following season and also the growth of the crop? So there was no significant difference among the different functional groups. As you can see here, these bars here represent the standard error, the variability among the different plots and among the different replicates, and it was quite high, and it's usually very high for wheat. So this was not unexpected, but it resulted in there being no significant differences overall, or very few among the different functional groups. So, but during grown after blend 2 here with the brassicas had lower wheat biomass than after after the next to it after blend 3. So this was not unexpected, because there would be fewer a lower seed bank carried over from the cover crop phase to the cash crop phase. So in terms of crop biomass, even though there were few differences when we conducted contrast analysis among the different groups, we found that the crop biomass in after the legumes, the monocultures, was higher. And again, this would not be unexpected because of the higher nitrogen level. And the in terms of yield, again, there weren't many differences, but when we did contrast analysis, we found that the yield of the durum was lower after the brassicas, circled here, than after the grasses or legumes, even though it doesn't really show very well here, but it is true when you do a statistical analysis and compare them in different ways. So in terms of protein, for those of you interested in a higher protein in durum, we found that the protein levels after the monocultures were the highest. And that of the functional groups, and it was also protein was also higher in the blend 3 dominated by legumes than in the other blends. So again, not unexpected. So therefore, as a summary, then durum wheat grown after the blend dominated by brassica had a lower wheat biomass than after the blend dominated by legume. And the highest crop biomass of durum was when durum was grown after the legumes. In terms of the yield, durum yield after the brassicas was lower than after the grasses or legumes. And that was, I didn't show you data on grain weight, but this is partly or mostly attributed to the fact that durum grown after brassicas had the lowest grain weight, and protein concentration was highest after the legumes. So in general, our projects have shown a potential for cover crops or grain intercrops, which I mentioned before that we have a very large project on that to successfully produce high amounts of crop biomass with low wheat biomass under very dry conditions prevalent since 2017. However, there is a low ability to convert simulates into grain when there is not enough soil water for grain filling. And this is what we call a low harvest index, which is the relationship between yield and crop biomass. So how much of the crop biomass translates into grain yield. So therefore, if grain conditions persist throughout the summer, any enhanced vegetative growth might not fully transfer to grain yield. And this is what has happened with some of our years and some of our trials. that we had a high crop biomass, but the differences were not that great for grain yield. So therefore, it's very important, especially when you're assessing the performance of different cover crops and following cash crop, that you measure the crop biomass, because it might not translate into grain yield that year, which might be very dry, but it might perform better the following year if the weather, if there is more moisture, which we're always hoping for. So what are the next steps in our cover crop research? We have this project now that we expect that it will be the letter of intent has been approved, and we expect that the actual proposal would be approved, and this is started in 2026. And basically it has the same or similar objectives as the previous cover crop work, but it has kind of a continuous rotation for 4 years. And this year we added also greenhouse gas emissions, which is a major component, and carbon sequestration, and this will be done by our colleagues involved in this project. We also added conventional trial of cover crops for in rotation with wheat. And in addition to that, we're also assessing the potential of integrating livestock in this project. And this is, I should mention that this is part of a multi-location throughout the country, and assessing cover crops for emissions, for greenhouse gas emissions. And the principal investigator, the one that coordinates its work, is Roland Krobel from Lethbridge, from the Lethbridge Research Center. But again, this project is across the country, and the data that we'll be able to gather will be quite significant and quite important for reduction of greenhouse emissions. So again, I just wanted to mention the research that we didn't mention here, or because of lack of time, and also other projects. You can find different articles on the Sask Organics website, or you can ask me directly, or Noe as well. And I just wanted to mention the latest peer review publications that are in press right now, but will be posted on the Agronomy Journal in the next few days. And it's in relation to intercropping of oat or mustard with legumes. And this is one of the papers, and the second paper is in regards to the cereal crop following organic intercrops and their respective monocrops. So and there are some commonalities between the intercropping work that we're doing and the cover cropping that I presented here, and or didn't present for brevity. Okay, so and one of the things that I wanted to mention briefly, that there were other important findings that we have reported and will continue being reported, and this has to do with endophytes, and this refers mostly to the intercropping trials, but also the cover crops. And endophytes are microorganisms that live within a plant, and without causing any disease. And in addition to their ability to control crop diseases, endophytes, such as the one that we have isolated from our organic trials, may increase crop performance by promoting plant growth and improving drought tolerance. And we have shown pictures about that, how mostly we did that work in a growth chamber, and how these endophytes were able to stimulate growth and resist drought conditions. So among the articles available on the Sask Organics site is this one on this biocontrol that I just mentioned, and also we published a paper a few years ago where these are actually pictures on the microscope where you can see the interaction between the endophytes and the pathogens. And the pathogens that we tested back then were Fusarium pathogens, responsible not only for Fusarium root rot, but also for Fusarium head blight. And in addition to that, but we haven't published it yet, we found that the same endophytes are able to control aphanomyces, which, as you likely know, is a very important disease of pulses everywhere, including the prairies. And I thought I would mention that significant findings such as this might only be identified and quantified under organic management. And the reason for that is, of course, that in conventional systems, you have synthetic chemical inputs that basically affect the survival and multiplication of those organisms. And another thing in relation to that I should probably mention, and that would encourage more people to practice organic management, is the fact that after more than 10 years now of having organic management in our lands, we are not able to isolate Fusarium pathogens. And this is quite significant. The only fungi that we have been able to isolate lately are saprophytes and fungi that are involved in biocontrol, but not pathogens. So anyway, so we have field days every year. You're welcome to attend. Except the only year that we didn't have any were during the pandemic, 2020 and 21. And in addition to touring our different field trials, we also have presentations in the afternoon on different topics, not necessarily related to our trials, but to work done by other people. So, okay, so what I wanted to mention briefly is this. I don't know how many of you are familiar that there's another round of a climate action fund, and EcoCert has sent me this poster. And this, there will be two streams, cover crops and or nitrogen management. And Noe is going to post the email where you can get more information. I believe that all the information and the required forms are not yet available, but EcoCert will have them ready on April 1st, which is when this program opens. So it's if you're not familiar with this fund, please read about it and contact EcoCert for more information. Do I have a few more minutes, Angelica?
I am not sure at this point if there's people with questions. Okay. Let me see.
Yeah, no, that's fine. I'll just mention this. I'll just mention this project, and it's a live and mulch project that has been very successful. And please contact us if you would like to get more information about it. Thanks a lot.
Okay, I think this is the end of our seminar at this point. If you have any more questions, as Dr. Fernandez has mentioned to you, you can send your questions to her technician. The email address is provided in the chat, and any more questions, you can also forward it to me. My email address is angelica@laraonline.ca. I would like to thank Dr. Miriam Fernandez for assisting this webinar. We are very happy to have you here. Even though it's virtually, we are looking forward to one of these days to come and see us in person. Hopefully that happens one day. And yeah, that's all I wanted to say at the moment. Is there anything else you would like to add, Dr. Fernandez?
No, not really. I just wanted to thank everybody for your interest in this topic. And as Angelica said, if you have any any questions at all, let us know, and we can send you not only a reply to your question, but also documents and information on our trials.
Okay, well, thank you so much. And yeah, so thank you so much for assisting. And I hope all of you have a great day. Bye.
Thanks a lot. Thanks.